double fertilization. So, in angiosperms, double fertilization takes place. It involves syngamy and triple fusion. What? Triple fusion. An important characteristic feature of angiosperm plants, double fertilization and triple fusion. Syngamy is the fusion of male and female gametes. Fusion of male and female gametes. That is syngamy. Whereas fusion of three nuclei is called triple fusion. Triple fusion. Syngamy results the formation of zygotes. Triple fusion involves formation of primary endosperm nucleus. Primary endosperm nucleus. It is triploid, it is diploid. So, we know the embryo sac having synergids and an egg cell at the micropylar end. Here you know the polar nuclei, the antipodals at the chalazal end. This is chalazal end. This is micropylar end. The embryo sac. Two polar nuclei. Central cell. This is antipodals. Egg cell synergids. This is pollen tube coming through tube nucleus, generating nucleus. And after pollination, the stigmatic region receives the pollen grain and their germination begins by the pollen grain and pollen tube passing through stigmatic tissue, style, then ovary wall, and finally reach up to the micropyle. And one of the synergids we know producing finger-like outgrowth called filiform apparatus. And the point of contact of pollen tube and filiform apparatus get dissolved and direct communication takes place between synergid and pollen tube through the filiform apparatus. The generative nucleus divides into two sperms. So these are sperm cells, sperms. This is entering into the synergids. Two nuclei entering into the synergids. One of them fuses with the egg. That is syngamy, formation of zygote. The other nucleus moves towards the polar nuclei to form, they fuse to form primary endosperm nucleus. And now the central cell becomes PEC. PEC means primary endosperm cell. Primary endosperm cell. Here fusion takes place. As a result, primary endosperm nucleus is formed. And so the fusion of male and female gametes resulting in the formation of zygote which is diploid, and the fusion of three nuclei, that means two polar nuclei and a sperm nucleus. First of all, the two polar nuclei fuse to form secondary nucleus or definitive nucleus which is diploid. Together with the sperm cell becomes triploid primary endosperm cell. Primary endosperm cell. And triple fusion results the formation of primary endosperm cell which is triploid and later becomes the endosperm. And so this is about the double fertilization. See, at a time two fusion takes place. Fusion between male gamete and female gamete, and fusion between the secondary nucleus together with the sperm. Primary endosperm nucleus, zygote cell, primary endosperm nucleus within the primary endosperm cell. Then the process of double fertilization in angiosperm was suggested by S.G. Navashi. Navashin suggested the double fertilization. So generally we can say the double fertilization in the angiosperms resulting in the formation of a zygote and a primary endosperm nucleus. Syngamy and triple fusion. See, post fertilization changes in a flower. Flower. Stigma. Decay of. Style. Decay of. Ovary becomes fruits. Ovary wall becomes fruit wall. Or pericarp. Ovary wall becomes fruit wall. The pericarp. Ovule. Seed. Integuments. Seed coats. Outer integuments. Testa. Inner integuments. Tegmen. Inner integument becomes tegments. Together constitute the seed coats. Then egg, nucellus become perisperm. Antipodals disintegrate. central cell PEC, primary endosperm cell, then polar nuclei, pen, egg, zygote. Egg becomes zygote. Synergids disintegrate. Synergids disintegrate. Then stamens folds off or decay off. Corolla folds off. Then calyx folds off or persist. Then flower stalk may become fruit stalk. These are the post-fertilization changes in a flower. So we know the stigma decay off, style decay off, ovary becomes fruit, ovary ball becomes fruit ball, ovule becomes seed, integuments become seed coats, outer integument testa, inner integument tegument, nucellus becomes perisperm, antipodals disintegrate, then central cell becomes primary endosperm cell. Primary endosperm cell. Primary endosperm cell. PEC cell. Polar nuclei. Pen, primary endosperm nucleus. Egg cell, zygote, synergids, disintegrate, stamens, folds off or decay off, corolla, folds off, calyx, folds off or persist. They remain there together with the fruits after fertilization. That is called persistent calyx. Flower stalk becomes fruit stalk. These are all the post-fertilization changes in a flower. Okay. Post-fertilization events in a flower. What are the post-fertilization events in a flower? There are 4 events, main events after fertilization or double fertilization. They are the formation and development of endosperm. The embryo. So, formation, development of endosperm, development of embryo, development of fruit, and that of seed. Or generally we can say the formation and development of endosperm, the formation and development of embryo, the formation and development of fruits. Formation and development of seeds. These are the main 4 events after double fertilization in angiospermic flower. Say once again, please listen. Formation of endosperm, formation of embryo, fruit, seed, which are all the 4 important events. Formation and development of endosperm. See, formation and development of endosperm. Endosperm is the nutritive tissue of the seeds. It is used for the development of embryo and seeds. Embryo and seeds. It is developed from primary endosperm cell. Primary endosperm cell, from primary endosperm nucleus, we can say it is developed from primary endosperm cell. PE cell, that means primary endosperm cell. In some seeds there is no well developed endosperm because it is consumed completely for the development of embryo and seeds, and that is why seed may have with endosperm or without endosperm. So, seed with endosperm. Endospermic seed. Or albuminous seed. Example, coconut, castor. Here the endosperm persists even after the fertilization and moreover to the seeds and their development. See, seed without endosperm is non-endospermic. Seed or non-albuminous seed albuminous seed or ex-albuminous seed. Seed without endosperm, non-endospermic. Example, peas, beans, groundnuts, where the endosperm is completely consumed, and that is why they are called non-endospermic or non-albuminous or ex-albuminous seeds. Then generally there are two types of endosperms. First is nuclear endosperm. Second is cellular endosperm. See, intermediate endosperm is also there. It is not yet given in your textbook. And so nuclear endosperm. Here the primary endosperm nucleus divides mitotically, forming numerous nuclei without followed by wall formation. That is example coconut water. Cellular endosperm, the pen divides mitotically, followed by wall formation. Example, corn kernel, coconut kernel. See, listen. Endosperm is the nutritive tissue of the seeds. It is used for the development of embryo and seeds. It is developed from primary endosperm cell. Seed with endospermic seed or albuminous seeds. Example, coconut, castor, etc., sunflower, etc. Then seed without endosperm, non-endospermic seeds or non-albuminous seeds. Ex-albuminous seeds. Example, peas, beans and groundnuts. See, there are two types of endosperms. Nuclear endosperm and cellular endosperm. In nuclear endosperm, the pen or primary endosperm nucleus divides mitotically, forming numerous nuclei without followed by the wall formation, cell wall formation. Then the example, coconut water. Cellular endosperm is here the primary endosperm nucleus divides mitotically, followed by wall formation. Example, corn kernel, coconut kernel, etc. See, this is the embryo sac. This is pen. This is pen. So the pen divides. Free nuclei. This is nuclear endosperm. But here this is zygote. Then the nucleus divides mitotically, forming number of, followed by wall formation, engulfing one nucleus within the cell. This is the two important different kinds of endosperms, that is nuclear endosperm and cellular endosperm. Nuclear endosperm present in coconut milk, coconut water, and cellular endosperm will be the kernel, thenga in Malayalam, you know. Yes. The embryo. The zygote develops into an embryo. Zygote develops to form an embryo. So zygote is diploid by the fusion of male gamete or sperm and the female gamete egg. So the mitotic division, continuous mitotic division helps to the formation of the mature embryo. See? This is the zygote. Before that, there are various steps for the fully developed embryo formation. First is proembryo. Zygote carries proembryo. Globular embryo. Heart-shaped embryo. Mature embryo. These are the four stages for the formation of a mature embryo from a zygote. Proembryo, globular embryo, heart-shaped embryo, then mature embryo. Heart-shaped embryo is also known as cord embryo. See, this is the zygote. This is the zygote. It produces two cells by the mitotic division. This is proembryo. It is for the embryo. The newly formed cell becomes proembryo. It develops to form the proper embryo or embryo proper. This is proembryo. And again, two mitotic division. So listen once again, the formation and development of embryo. So embryo is developed from the zygote. So for the formation and development of embryo, there are four steps: proembryo, globular embryo, heart-shaped embryo, mature embryo. This is the zygote. The zygote divides into two, forming a terminal cell and a basal cell, the proembryo. This is proembryo for the embryo. Second stage is the division followed by the formation of globular embryo. That is, basal cell divides into two. This cell divides into two. Terminal cell or free end cell periclinally and anticlinally divides more a globular mass of tissue called globular embryo. So again division takes place from each and every cells, so that the globular embryo becomes a cone-shaped appearance or a heart-shaped appearance, and hence the name heart-shaped embryo or cord embryo. So this embryo becomes curved towards one side, and the other cells become disintegrated, and it becomes the suspensor. Suspensor suspend the mature embryo. Suspensor suspend the mature embryo. This is curved back and like this. And the seed development of the seed also, the endosperm also developed during these stages, and the seed keeps the embryo safely inside them. And when the favorable conditions comes, the embryo develops to form a new plant, and that is about 4 stages: proembryo, globular embryo, heart-shaped embryo, mature embryo. Then typically, an embryo has 2 distinct parts, namely an embryonal axis and two cotyledons, an embryonal axis and 2 cotyledons in dicot embryo, single cotyledon in monocot embryo. That is why, depending upon the number of cotyledons, the embryo is of 2 types. In turn, plants are also of 2 types. Dicotyledonous plants having 2 dicots in their embryo. Monocotyledonous plants, only a single cotyledon in their embryo. See, like that. And that is why a typical embryo contains an embryonal axis and 2 cotyledons in dicots. Then the mature embryo. This is a mature embryo, dicot embryo. These are the cotyledons. There is a small protrusion in between the cotyledons, that is called plumule, which later becomes the shoot system, plumule. So the cylindrical structure, this is the embryonal axis. This is the embryonal axis having epicotyl, hypocotyl, radicle becomes the root system. And this is the root cap. So typically we can say the part of embryonal axis above the plumule is called a epicotyl. That means while we examine, suppose a new plant, cotyledon, plumule. This is cotyledon. This is cotyledon. This is epicotyl. This is hypocotyl. And the middle part is mesocotyl. The cotyledon's point of attachment of cotyledon to the mesocotyl, and above it is epicotyl, below it is hypocotyl. And hypocotyl develops to form the root or radicle and root system that you can see later. That you can see later. So this is plumule. This is plumule. The two cotyledons, since it is dicots, and embryonal axis. The whole cylindrical part is embryonal axis. This is, and having the part above the plumule is epicotyl, and the part below the part of embryonal axis below the cotyledon is called actually the hypocotyl, radical plumule. So the figure you have to know, the figure is given, you have to name the parts of the dicot embryo. This is dicot embryo. Typically an embryo contains an embryonal axis and two cotyledons in dicots. And this is the figure of, so this in your textbook it is given in this manner. This is mature embryo. To one side, opposite sides, opposite sides. So this is then mature monocot embryo. See, this is a monocot embryo. And the figure is given. and then you have to label them, and the parts. This is the flattened single cotyledon, that is called scutellum. Single shield like flattened cotyledon of monocot embryo is called scutellum. It is in one side, towards one side of the embryo, and this is the embryo having plumule, radicle, root cap, coleorhiza. This is parts concerned. Scutellum, this is coleoptile plumule sheath. This is plumule. This is epiblast, the leaf primordia which will be there. This is radicle. And this region, root cap, and this is coleorhiza, radicle sheath. This is monocot embryo. Listen please. The not shaded scutellum. What is scutellum? Single shield like flattened single cotyledon of monocot embryo. What is scutellum? Shield like flattened single cotyledon of monocot embryo is called scutellum. Then coleoptile, plumule sheath of monocot embryo. Plumule sheath of monocot embryo is called coleoptile. Coleorhiza, that is Radical sheath of monocot embryo is coleorhiza. So I have to remember rhizoid of roots. Rhizoid or not. So distinguish between coleoptile and coleorhiza. Maybe asked. And then you have to remember rhiza stands for rhizoid root radical. So radical sheath of monocot embryo is coleorhiza. Coleoptile, that is plumule sheath. So both plumule sheath and radical sheath distinctly present in monocot embryo. And so that is why, so scutellum, coleorhiza, coleoptile, which are all, then epiblast is actually a small protrusion which becomes the leaf primordia. And so that is about what the monocot embryo. See, dicot embryo, monocot embryo. Then next is, these are all about the events, that is the formation of endosperm and formation of embryo, formation and development of embryo. The next is fruits. Already learned in your morphology in detail. Formation of fruits. Fruit is developed from so fertilized ovary means ovary after fertilization becomes the fruits. It has a covering called fruit wall, also known as pericarp. Also known as pericarp. The fruit wall of simple fruits has three distinct layers. Namely, epicarp, mesocarp and endocarp. Fruit encloses, fruit wall encloses the seeds. Encloses the seed or seeds. See, formation of fruits. Fruit is developed from the fertilized ovary. It has a covering called fruit wall, also known as pericarp, developed from the ovary wall. The fruit wall of simple fruit has three distinct layers: epicarp, mesocarp and endocarp. Endocarp encloses the seeds. Suppose Suppose longitudinal section of a mango fruits to identify the LS of a mango fruits. See, this is fruit stock. Epicarp, skin of fruit wall. This is thin, membranous and fleshy mesocarp which is edible. And this is endocarp. These three constitute pericarp or fruit wall. And within it the seed present. This is seed is there. A simple fruits. A simple fruits showing the pericarp. So fruit is developed from the fertilized ovary. It has a covering called the fruit wall, also known as pericarp. The fruit wall of simple fruit has three distinct layers: epicarp, mesocarp and endocarp. The fruit wall encloses the seed or seeds within it. A simple fruit showing the pericarp. Epicarp, the outermost covering, thin, membranous, maybe. Mesocarp, fleshy or fibrous. Endocarp, also fibrous and thin. Then endocarp encloses the seeds. So this is the longitudinal section of a mango fruit. Okay, then remember, developed from true fruit is developed from monocarpellary superior ovary. Fruit is generally divisible into true fruit, false fruit, pseudocarpic fruits, fruit that will not developed from fertilized ovary, but other part other than the ovary becomes the edible part, and such fruits are said to be false fruits. Say for example, apple, strawberry, then cashew fruits. Apple, strawberry, cashew fruits. In apple and strawberry, the edible part is thalamus. Here in cashew fruits, fruit stalk is edible. Means this is the nuts. Cashew nuts, and this is stalk. This is edible part. So here thalamus is edible cashew fruits. A simple fruit is developed from multicarpellary syncarpous ovary or monocarpellary unilocular ovary. Based on the nature of the pericarp or fruit wall, the simple fruit is divisible into dry fruit and fleshy fruits. In dry fruit, the pericarp becomes fleshy when the fruit attains maturity, and become dry when the fruit attains maturity, and here it becomes fleshy when the fruit attains maturity. So dry fruits may be dehiscent, but they dehisce. Dry pericarp dehisce to remove or to the dispersal of seeds. Dehiscent and indehiscent. Indehiscent, so paddy, indehiscent. Dehiscent, mustard, or we can say pea. This is paddy. And fleshy fruit is tomato. This is berry fruit, drupe, coconut. See, among them you have to go through the drupe, developed from monocarpellary superior ovary. Developed from monocarpellary superior ovary or unilocular superior ovary. So a drupe is developed from, example, coconut, mango. Mango is a fleshy drupe. This is fibrous drupe. Fleshy drupe. Because the mesocarp is fleshy and edible. It is fibrous. Mesocarp is fiber for making coir. So like that. Agreed fruit. Anona. Multicarpellary apocarpous ovary. This is syncarpous. Multicarpellary apocarpous ovary becomes Anona squamosa. Multiple fruit pineapple, jackfruit. Here an inflorescence become fruits. That is pineapple and jackfruit. Example for multiple fruit or compound fruits. Multiple fruit or compound fruits. And so that is about a general account of fruits. See, fruit is in most of the plants, the fruit is edible as it is rich in nutrients. Okay, then false fruit, apple, strawberry, cashew fruit. It is not developed from fertilized ovary, but the edible part is the part other than ovary. That is why pseudocarpic. So false fruit. Fruit thus divides into true fruits and false fruits. Seed, the fourth one. Third is fruit, fourth is the events of fertilization, that is seeds. Seed is the fertilized ovule. Fertilized ovule. It has a seed coat. Seed coat contains an outer coat, testa, and an inner coat, tegumen. The seed coat contains an outer coat, testa, and inner coat, tegumen. They constitute the seed coats. The scar on the seed coats is hilum, the funicle connecting to the ovule, body of the ovule. Then the nutritive parts, part or nutritive tissue of the seeds. Endosperm. Seed with endosperm, formerly given. Seed with endosperm, endospermic seeds. Seed without endosperm. Non-endospermic seeds. Then seed without endosperm, non-endospermic seeds. Most important part of a seed. Embryo. The most viable seed having 10,000 years old, seen in Lupinus articus. Viable seed having 2,000 years old. 2,000 years old. Phoenix ductilifera. Date palm according to the archaeological excavation. Archaeological excavation. According to the archaeological excavation, the most viable seed having 10,000 years old seen in Lupinus articus in Arctic region. The viable seed having 2,000 years old, Phoenix ductilifera or date palm, seen in the King Herod's palace, seen in the King Herod palace near the Red Sea. Then smallest seed present in orchids. Largest seed double coconut. Largest seed present in double coconut, cycas, etc. Then the small seeds that present in parasitic plants like Robang and Striga. So small seeds that present in parasitic plants like Striga and Urobank, they are actually parasitic plants. Small seeds are present. The residual persistent endosperm, residual persistent endosperm, perisperm. Perisperm, scar, etc. See, points to remember. Seed is the fertilized ovule. It is the end product of sexual reproduction in flowering plants. Sexual reproduction in flowering plants. Remember please. So points to remember in connection with the seeds. Seed is the fertilized ovule. It has a seed coat. It has a coat. The seed coat. The seed coat contains an outer coat testa and inner coat tegumen. They constitute the seed coat. The scar on the seed coat is hilum. The nutritive tissue of the seed endosperm. Seed with endosperm, endospermic seed or albuminous seed. Seed without endosperm, non-endospermic or ex-albuminous seed. The most important part of a seed, embryo. The most viable seed having 10 years old, seen in Lupinus articus. The most viable seed having 10 years old, Lupinus articus, seen in Arctic region. Then viable seed having 2000 years old, Phoenix dactylifera or date palm. Then smallest seed present in orchids, largest seed present in double coconut, then cycas. Cycas is not a flowering plant. See, the small seeds that present in parasitic plants like striga and orobank. Then the residual persistent endosperm of the seeds, perisperm. Perisperm is there in cedium, seeds of cedium, and pepper, pepper, etc. So these are all some important key points in connection with the seeds. Then significance of the seeds. See, fertilized ovule, seed coat, testa tegumen, hilum, endosperm, endospermic seed, non-endospermic seed, most important part of a seed, then most viable seed, 10 years old, Lupinus articus, viable seed. According to the archaeological excavation, this is suggested because archaeological excavation revealed that the most viable seed they discovered in Arctic region, the plant is called Lupinus articus, 10 years old. Sorry, 10,000 years old, not 10 years. For that 1 minute. years old. Yes, 10,000 years old. Phoenix dactylifera. This is Lupinus articus. Then smallest seed orchids, largest seed double coconut. Say a small seed that present in parasitic plants like striga and orobank. The residual persistent endosperm called perisperm, seen in pepper. Guava, pepper, you have to remember pepper only. Then 5 important points to remember about the seeds. This is the general account of seeds. Important significance of the seeds. Seeds are rich in nutrients. Hence it is edible. It is the basis of agriculture. Since seed is dehydrated, it can store and transport distant places. It is used in the crop improvement program. See, 5 important key points. Seeds are the end product of sexual reproduction angiosperms or flowering glands. Seeds are rich in nutrients, hence it is edible. It is the basis of agriculture. Since seed is dehydrated, can store and transport in distant places. It is used in the crop improvement program. So, qualitatively and quantitatively high yielding varieties of plants can be hybridized. Plants can be produced having disease resistant capacity, pest resistant capacity, and stress resistant capacity. The new plants thus produced can be distributed each and everywhere. And so, for germination, seed needs water. For germination, water is needed. Okay, water and oxygen is needed. These are the important. Next is a monocote seeds with embryo. Fruit wall and seed coats. This is this is monocot embryo with the seeds. So monocot embryo is shifted to one side of the endosperm. Remember please. Fruit wall and seed coat is there. Into that there is a protein layer called aleuron layer or protein layer. Protein coat or layer. The protein coat of wheat is gliden. Remember please, gliden. That of wheat, gliden. Protein coat or aleuron layer of wheat is called gliden. This is endosperm, the edible part. And embryo is shifted to one side. Monocot embryo already learned. This is coleoptile, that is plumule sheath. Plumule, scutellum, radicle, coleorhiza. These are all the parts of a typical monocot seed. See? And then apomixis and polyembryony. Apomixis and polyembryony. So listen. The seeded plants are produced without fertilization. Is called apomixis. The plants through apomixis is called apomict, called apomict. Such plants are seen in Asteraceae and in some grasses. Then occurrence of more than one seed, occurrence of more than one embryo in a seed. Is called occurrence of more than one embryo in a seed is called polyembryony. Occurrence of more than one embryo in a seed is called polyembryony. Example, citrus, mango. Citrus and mango. Development of fruits. Without fertilization. Development of fruit without fertilization is called parthenocarpy. And such fruits are called parthenocarpic fruits. Example, banana. and that is about sexual reproduction in flowering plants. ശ്രദ്ധിക്കുക. So all of you listen. Monocot seed with embryo having fruit wall and seed coats, aleuron layer or protein layer, endosperm the edible parts, coleoptile the plumule sheath, plumule, scutellum, radicle. The figure is given and so you have to name the parts. Or 4 or 5 names will be given based written as A, B, C, D, 2 marks. So somebody sometimes may ask us copy the diagram then label it, 3 marks. So here aleuron layer, the layer which separates the monocots, that is also aleuron layer. The protein coat of monocot seed, aleuron layer. The layer which separates the endosperm and embryo in monocots, aleuron layer. These are the 2 important questions. Others are given. Coleorhiza, the radicle sheath. Then apomixis and polyembryony. The seeded plants are produced without fertilization is called apomixis. The plants through apomixis is called apomicte. Such plants are seen in Asteraceae and in some grasses. Occurrence of more than 1 embryo in a seed is called polyembryony. Example, citrus and mango. Development of fruits without fertilization. Development of fruit without fertilization is parthenocarpy, and such fruits are called parthenocarpic fruits. Such fruits are called parthenocarpic fruits. And so parthenocarpy, polyembryony, coleorhiza, coleoptile, aleuron layer, then scutellum, epiblast, drupe, then perisperm, pericarp, which are the key points to remember, not only for theory but for your entrance or competitive examinations, whatever may be. And so these are all about what the sexual reproduction in flowering plants. Okay, then next lesson, that is on the next class.